Hello everyone, I am Qazi Faruddin. In continuation of my previous lecture where I had discussed absorption and translocation of water in plants. So in this lecture I am going to discuss in detail the final journey of water from leaf to atmosphere. So it's very interesting to know that how water is lost from the plant surface. So as I had discussed in my previous lecture, the main driving force for movement of water from soil to leaves or from leaves to atmosphere is transpiration, and water generally moves through the xylem at the rate of only 4 millimeter per second during fast evaporation. Also, one carbon dioxide molecule is gained for every 400 to 600 water molecule lost. So you see, for gaining one molecule of carbon dioxide, how much water is lost? About 4 to 600 water molecule is lost. So it's very interesting to know that sunflower plant imbibes how much imbibes and perspire about 17 times more water than a human at every 24 hours. Since an equal quantity of water must be absorbed from the soil to make up for the water loss through the transpiration. However, apart from the major water loss caused due to the transpiration, it is also beneficial for plants in many ways, like rapid evaporation of water from the aerial parts of the plant through transpiration basically reduces the temperature of the leaves and thus prevent them from excessive heating. Additionally, we have already discussed the primary benefit of transpiration, that is development of suction pressure, which facilitate the uptake of water and nutrients from the soil. Therefore, many workers like Curtis, as I have already mentioned, call the transpiration as necessary evil. So in this given figure, you just see that water is drawn from the xylem into the mesophyll cells, which are green in color, and it reaches into the mesophyll cell wall during the flow of water from the leaf to the atmosphere, where it evaporates into the air spaces of the leaf. Now the water vapor is regulated by gradients in water potential and leaves the leaf through the stomatal pores, as you see in this figure. On the other hand, vapor phase transport occurs by diffusion, that is the concentration gradient of water vapor governs the last segment of the transpiration stream. Depending upon the plant surface, transpiration could be of three types. Stomatal transpiration, you see that small openings are present on the leaves, and through these minute pores transpiration takes place, and it constitutes about 85 to 90% of water lost by plant. Then second is cuticular transpiration. Here water vapor is lost to the cuticle, but the amount of water loss is generally very small and contribute about 5 to 10% of the total transpiration. This type of transpiration depends upon the thickness of the cuticle and presence or absence of wax coating on the surface of the leaves. Mainly in xerophytic plants, generally these plants have very thick cuticle and wax coating on the leaves and stem in order to basically check cuticular transpiration. Then third is lenticular transpiration. Generally water evaporate through these certain small openings present on the older stem. These openings are called lenticels, and the transpiration that takes place through these lenticels is known as lenticular transpiration. Huber basically noticed that in some plants water loss by lenticular transpiration was about 0.1% of the total transpiration. What he further noted that coating the bark of the trees reduced the loss of water by 20% from the total bark surface, indicating that some water loss was taking place to these general surface of the bark. Now the rate of transpiration is generally affected by uh, many factors, mainly the plant water status, plant anatomy, or the environmental factors. Among the environmental factors, most important are light, temperature, humidity, wind velocity, along with others. So here we will discuss each one of them one by one and study their effect on transpiration individually. But we should keep one thing in mind, that these factors influence each other in natural habitat and consequently affect the rate of respiration. First is, you see that temperature. If all the other factors are kept constant, a rise in temperature generally increases the rate of transpiration, as mentioned earlier. As I mentioned earlier, more the difference in water pressure, higher is the rate of transpiration. So an increase in the temperature increases the kinetic movement of water molecules. Therefore, the difference in vapor pressure of the leaf and outside the atmosphere also increase. Hence, faster the transpiration rate, but if plant faces a temperature rise above 35 degrees C, generally stomata close. How? So that if stomata close, transpiration won't take place, therefore the rate of transpiration decreases beyond a certain temperature. Now, what is the effect of wind velocity and rate of transpiration? What happens generally during high wind velocity, stomata tend to close as a protective mechanism to avoid desiccation and water loss. So this reduces the transpiration rate. So when wind velocity is low, it promotes the transpiration because gentle wind disperses the air surrounding the leaf, which in turn reduces the water pressure in the intermediate vicinity of stomata. So this decrease in vapor pressure facilitates the movement of water vapor from the leaf surface into the atmosphere, thereby increasing the rate of transpiration. Now, what is the effect of light on the rate of transpiration? So light has a very significant influence in controlling the movement of stomata. Generally, stomata open in light and close in darkness. So this is the general condition. But the rate of transpiration is also influenced by light intensity. What happens? Higher light intensity typically leads to an increase in transpiration rate. This is because light stimulates stomatal opening, allowing for greater water loss through transpiration. Additionally, light triggers photosynthesis, which generates energy required for the opening of stomata and the movement of water molecules from the roots to the leaves. However, excessive light exposure can lead to stomatal closure and a decrease in transpiration rate as a protective mechanism to prevent excessive loss of water. Therefore, while light generally promotes transpiration, the response may vary depending upon the intensity and duration of light exposure, as well as the type of plant species and environmental condition. Now, what is the effect of humidity on the rate of transpiration? You see that any sort of decrease in amount of water in atmosphere creates a sort of gradient that allows water to move from leaf to atmosphere. The higher the relative humidity, the more moisture in the atmosphere, which lowers the driving force for transpiration. Opposite to it, the lower relative humidity, the less moisture there is in the atmosphere, which increases the driving force for transpiration. Now, what are the ecological adaptations to minimize transpiration? So the rate of transpiration could be controlled as per the growing condition of the plant. Therefore, plants employ various strategies and adaptations to reduce loss of water through transpiration. So the presence of sunken stomata, thick cuticle, and presence of stomata on the lower side of the leaf epidermis are considered as strategies and adaptive mechanism modified by the plants. So you might have observed the plants growing in desert conditions face a sort of water crisis. So in such condition, what happens that the plants need to somehow limit the rate of transpiration, and the desert plant generally reduce surface area of the leaf, possess sunken stomata, have thick cuticle, and generally stomata are open during night. And in addition to this, such plants have modified special water storage capacity, root structure, and generally carry out C4 photosynthesis. So these are the measures adapted by the plant to minimize loss of water through the process of transpiration. Now, there are certain chemicals which are generally used to reduce the loss of water from the plants through the transpiration, and these are known as antitranspirants. And these antitranspirants are useful for reducing the transplantation shock to the plants, raised generally through tissue culture or raised in nursery. So an antitranspirant must be non-toxic and affect only stomata and not the plants. So in addition to it, antitranspirant should not delve to cause permanent damage to stomatal movement. It should be temporary. The closure should be temporary. So there are 4 types of antitranspirants generally used for reducing the rate of transpiration. One is stomatal closing type. So such kind of antitranspirant which close the stomata like some of the fungicides, namely phenyl mercuric acetate, and herbicides like atrazine, they are stomatal closing types of antitranspirant. Then another category, next category of antitranspirants is film forming types. So basically such kind of antitranspirants form coating on the surface of the leaves and blocks the stomatal pores and results in reducing the rate of transpiration. And the materials could be like plastic or waxy materials. They come under this category of film forming types of antitranspirant. Now third type of antitranspirant, reflectance type. So these are the materials basically which form a white coating on the leaves and increase the leaf reflectance. For example, material like hydrated lime, kaolin, calcium carbonate, magnesium carbonate, and zinc sulfates, these are frequently used to reduce transpiration. Now the last category of antitranspirant is growth retardant. So you might have heard about very interesting plant growth hormone, abscisic acid. This is a class of plant growth hormone which could also be used to induce closure of stomata. And other chemicals like psychosil could be exploited to reduce shoot growth and increase root growth, thereby making the plants to withstand under drought stresses. So it could be also utilized to improve the water status of the plant. Now, what are the stomata? The green gatekeepers for gas exchange. So you see in this figure, this is on the left side, this is the stomata of corn showing dumbbell shaped guard cells, whereas this is the stoma showing of Tedescantia zebrina. This is a dicot, and you see that through these stomata, this comprise about 15 to 40% of the total leaf volume. However, the stomatal pores make up just 1% of the total surface area. So it is responsible for how much? More than 90% of the water loss due to transpiration. So the wall of the lining of the pore is generally thicker than the outer wall of the guard cells, which are thin generally. So, I will discuss in my next slide that how the stomata are radially controlled. So, a fully open stomatal pore measures about 5 to 15 micrometer wide and is about 20 micrometer long. So, just I mentioned that the guard cells are bean shaped in dicards, whereas in dumbbell shaped in monocards, that evident in this figure. Moreover, the number, size and distribution of stomata can vary among different plant species and can be influenced by environmental factors such as light intensity, humidity and temperature. So, basically what this stomata do generally? Stomata regulate the equilibrium between the intake of carbon dioxide and loss of water by regulating their movement. Now, what causes opening and closing of stomata? That is very interesting to know. What happens during daytime when photosynthesis takes place? Generally CO2 concentration drops and pH increases to 7, and in this condition malic acid is formed in guard cells. This malic acid then breaks into malate and hydrogen ion. These hydrogen ion moves out of the guard cells with the help of ATP driven pumps and pass to subsidiary or epidermal cells. Now, what happens once these protons pass out of the guard cells? In the next move, potassium from subsidiary or epidermal cells move into the guard cells where they react with malate and form potassium malate. So, this potassium uptake is generally balanced by uptake of colloid ion into the guard cell. So, this lead to increase in the solute potential of the guard cells. Hence, this lowers the water potential, which causes endosmosis, and the guard cells become turgid and causes opening of stomata. So, what happens during opening of stomata? This proton pump plays a very crucial and significant role in controlling the turgor of guard cells, with potassium playing a crucial role in decreasing the osmotic potential up to how much? 2 megapascal. So, stomatal opening can be stimulated by using certain fungal toxins like fusicocaine, which causes an active proton extrusion necessary for stomata to open. Also, what has been noticed that if proton pumps are inhibited, stomatal opening cannot take place even in the presence of blue light, which has been reported to induce a very strong opening, stomatal opening. Now, just opposite to it, what happens during night? When photosynthesis stops and CO2 concentration increases, so at this juncture ABA is released to stop potassium exchange and efflux of potassium and colloid ions from guard cells takes place. Then again proton flow back from epidermal cells into the guard cells, due to which pH of the guard cells decreases to 5 and further malate is converted into starch. This leads to a decrease in solute potential and generally exosmosis is observed and guard cells become flaccid and stomata close. This is known as proton transport theory, and at present this theory is most acceptable theory for explaining the regulation of stomatal movement. Now, just I mentioned, so this is on the left side you see dicart stomata, and on the right side monocart stomata. So, you see that during the stomatal opening microfibrils play an important role, and these are arranged in radial pattern around the guard cells, and these are known as radial myceliation. So when water enters in the guard cell, it basically primarily increases their length rather than diameter due to the presence of these radiating microfibrils. So this expansion exerts pressure on the microfibrils, causing them to pull on the inner wall of the stomatal pore, leading to opening of the stomata. Now, as I mentioned that stomatal movements are influenced by number of external and internal factors. So the environment surrounding the plants is very dynamic and keeps changing. So in response to these factors, stomatal apertures also adjust accordingly. So generally what happens is environmental signals are generally perceived by the guard cells, and these signals are transduced into ion fluxes as discussed earlier, leading to a change in the turgor pressure of the guard cells. So some of the important environmental factors affecting the stomatal movements are discussed. First is, you see, light. So in most of the plant, and just I mentioned that stomata are open in the light and closed in the darkness. So stomatal movements are basically influenced by both intensity and quality of light. The light intensity required for the stomatal opening is quite low, and even in some plants, stomata open even in moonlight. So as far as different lights are concerned, both red and blue light are very effective in stomatal opening, although blue light is slightly more effective in causing stomatal opening. However, what is the case in succulent or chyme plants? So the stomata open in such plant during night and remain closed during daytime. For example, Apensia, agave, aloe, kalanch, pineapple. These are the few examples of chyme plants. So this adaptation of chyme plants lead to carbon dioxide absorption during night time when transpiration stress is low. So the effect of light on stomatal movement is independent of photosynthesis. Generally, stomata respond to light even in the leaves where photosynthesis has been reduced to zero by the application of certain photosynthetic inhibitors like cyanazine. But one of the interesting fact I would like to share that blue light has direct effect on the opening of stomata independent of CO2 concentration. Like in Allium cepa, guard cell swells in the presence of potassium when blue light is generally exposed. Blue light enhances the proton transport, leading to proton gradient, which in turn causes the opening of potassium channels. Red light also generally stimulates stomatal opening, but blue light is 10 times more effective than red light. Now come to another factor which is also controls the stomatal movement. So what happens in most plants, an increase in temperature induces the stomatal opening. But what happens, the temperature above 35 degrees C induces stomatal closure. However, to ensure gas exchange at high temperature, stomata open during night time. And here it is worth to mention that temperature has direct effect on stomatal movement. It affects the balance of respiration and photosynthesis. Whenever there is increase in temperature, it leads to rise in respiration rate. Due to high rate of respiration, the level of CO2 increases and it causes closure of stomata. Contrary to this, high temperature induces stomatal opening in some plants to increase the transpiration rate. So what is the purpose of increasing the transpiration rate with the rise in temperature? That is to generate cooling effect, or you can say in order to maintain temperature of the leaf. Now next is, what is the impact of atmospheric humidity on stomatal movement? So humidity also affects the opening and closing of stomata in plants. what happens when the air is dry, or you can say that having low humidity, plants tend to lose water more quickly through the transpiration. In response to this, stomata may partially close to reduce water loss and conserve moisture inside the plant. Conversely, when the air is humid, or you can say that high humidity, so there is less risk of water loss through transpiration because the air is already saturated with moisture. So in such condition, stomata may open to wider to facilitate gas exchange for photosynthesis, as there is a need to conserve water. So it could be mentioned here that low humidity typically leads to stomatal closure, while high humidity promotes stomatal opening in plants. So what is the impact of CO2 concentration on stomatal movement? So it is mainly the internal level of carbon dioxide which plays a pivotal role in opening and closing of stomata. So in most plants, low carbon dioxide concentration in intercellular spaces causes the stomatal opening during night time, whereas high concentration of CO2 in intercellular spaces causes partial closure of stomata during daytime. Once stomata close, external carbon dioxide concentration does not affect the stomatal movement. High level of intracellular CO2 induces closure of stomata in the same way as abscisic induced stomatal closure. Now, what is the impact of oxygen on movement of stomata? Generally, oxygen is essential for opening of the stomata. Generally, ATP is produced through aerobic respiration and essential for opening of the stomata, and ATP is required to power the activity of potassium ion and proton pump. These are located in the guard cells of the stomata. These pumps actively transport potassium ions into the guard cells and from surrounding cells, creating a higher concentration of potassium ions inside the guard cell. So this increase in potassium ions inside the guard cells create an osmotic gradient that causes water to move into the guard cells, leading to their swelling and ultimately resulting in opening of the stomata. Now, next factor that affects the movement of stomata is availability of water. Sometimes what happens, due to water stress, the guard cells loses more water as compared to its intake. It loses its turbidity and stomatal closure takes place. This is called hydropassive closure of stomata. On the other hand, stomata also experience ABA mediative hydroactive closure. So another factor, hydrogen ion concentration, that could be measured in terms of pH. So the effect of pH on stomatal opening and closing is not direct and well understood. However, changes in the pH can indirectly influence the stomatal behavior through their effects on ion concentrations and generally enzyme activity and inside the guard cell. In general, guard cells which regulate stomatal aperture contain various ions and channels and transporters, and these are very sensitive to changes in the pH of the guard cells. These channels and transporters play a role in controlling the movement of ions such as potassium, chloride and hydrogen ions, which in turn affect the turgor pressure and osmotic potential of the guard cells. So generally findings suggest that acidic conditions or low pH may stimulate the activity of proton pump in the guard cells, leading to accumulation of hydrogen ion inside the cells and subsequently activation of potassium channels. This can result in an influx of potassium ion into the guard cells, causing them to swell and stomata open. Now, what are the impacts of growth hormones on stomatal movements? Generally, cytokinins are well established to stimulate stomatal opening, whereas ABA induces stomatal closure. So, ABA is another plant hormone that induces the closure of stomata and often referred to as stress hormone, because generally the level of ABA increases in response to various environmental stresses like drought, high salinity and low temperature. When ABA level rises, it triggers a signaling pathway that leads to closure of stomata, helping to reduce water loss from the plant and prevent dehydration in the plant. Now, how mineral status affect the movement of stomata? As you know that stomatal opening depends primarily upon the availability of potassium ions from the adjacent epidermal cells or subsidiary cells. So, a deficiency of phosphorus and nitrogen also slows stomatal opening, controls stomatal opening. Several other minerals are also essential for stomatal movements like magnesium and calcium. So, these environmental factors basically interact to regulate stomatal movement, ensuring an optimal balance between water conservation as gas exchange for photosynthesis even in fluctuating environmental conditions. So, thank you very much for attending this lecture. We have learned that how do stomata play a crucial role in loss of water from the leaf surface and how water is transferred from leaf surface into the atmosphere and various factors that governs the movement of stomata. That these aspects I have discussed in detail. Thank you very much.